Okay, so now that we have discussed how action potentials are generated and how they move down the axon membrane, either by saltatory or continuous propagation, at some point this action potential is going to reach the end of the axon at the synaptic terminal. So at that point the impulse has to move to either another neuron or to a muscular cell. So how does this actually happen? How does this action potential move from one neuron cell to another? Now, there's a couple of things you need to keep in mind. The neurons or the neuron and the muscle are not in direct contact with each other. They actually don't physically touch. In addition, if you're thinking like an electricity type of thing where it would just kind of spark over, that doesn't happen either. The impulse does not jump across and kind of like an arc, like if you have an arc of electricity between two wires or something like that. It doesn't work that way. How this transmission happens is chemically, and this is located at a place in the gap called the synapse. The synapse occurs between the synaptic knob of one neuron, its axon, and the dendrite of another nerve. Now, communication only happens in this one direction from the axon to the dendrite. Now, if you take a look at your packet at this point, if you flip actually, I think, to the next page, you'll see a diagram that kind of looks like this. All right. You actually have 4 of them, and I'm going to kind of walk you through how we're going to be diagramming this. The first thing you want to do is fairly close to your little synaptic knob there, you want to just draw a straight line down. Your papers are oriented landscape, so you can actually draw this the little diagramming that we need to do on one side, and you still have plenty of room on the other side to list the various steps that we are going to go through. So the first part of what we are going to do is label the first diagram so that we kind of know the pieces and parts that are involved in this process. First, the little knob there represents something we call the presynaptic neuron, in other words, the one before the gap. And on this side of the line would be the dendrite, and that represents the postsynaptic neuron. Inside the knob of the presynaptic neuron, we have little containers, which are called vesicles. And those blue triangles inside the vesicles represent neurotransmitter, which is a special chemical that's involved in this process. On the postsynaptic neuron, the line there represents the membrane of the postsynaptic neuron. We actually have special structures called receptors. Now, no matter how you draw these, the shape of the receptor end on the synapse side needs to be able to match up the shape of the neurotransmitter, because eventually they're going to fit together. The space in between is referred to as the synapse or the synaptic cleft. So that's all we're going to put on the first diagram. Now what we're going to do is we are going to actually move on to the second diagram. So I want you to set it up so that it looks like this. All right. Now, first step, step 1, and I don't have it actually listed numbered here because with the presentation I try to make it so that things will fit. The first step is that the impulse arrives at the synaptic knob of the presynaptic neuron. That leads to step 2, which is where we have calcium existing in the synaptic cleft, actually coming into the synaptic knob. Now, this intake of calcium causes the vesicles to move to the membrane, just like that. Okay, so they slide up to the membrane. Now, I know I can animate things, and you guys can't on that diagram. So what I would do is I would simply, obviously, leave those there and just draw arrows showing that they are moving to the membrane. And that is the end of that diagram. We're only going to put those 3 steps on there. So on your third diagram, you want it to start with a setup that looks like this. So our fourth step is that the neurotransmitter diffuses across the cleft. And that takes us to our 5th step, which is it binds with the receptor. So what you want to do is you want to take your pencil and actually draw an arrow running from the neurotransmitter directly to the receptor, and that will represent both steps 4 and steps 5. that neurotransmitter binding with the receptor causes sodium channels, this is step 6, to activate. So sodium moves through the channels on the postsynaptic neuron, and depolarization to threshold occurs, and we get a new action potential that is generated. And that is the end of our third diagram. Now in our fourth diagram, we have the setup that looks like this, with the vesicles with the neurotransmitter there in the presynaptic neuron, and the neurotransmitter still here attached to the receptors. Up until this point, we've pretty much just moved that impulse, but now we need to reset everything so that we are ready at the synapse for another impulse to come through. The first part is that the enzymes break down the neurotransmitter. Now again, on my PowerPoint here, I can actually make them disappear. What you could do is just draw X's over them, just showing that they're going away. Now, these pieces remain in the synaptic cleft, and they actually get reabsorbed by the knob and made into new neurotransmitter. That is because we've invested a lot of metabolic capital, so to speak, in producing this neurotransmitter, and we don't want to waste it. So that's the process at the cleft. So what exactly are these things called neurotransmitters? Well, let's go through some of the common ones. The two most common ones that we have in the body are acetylcholine and norepinephrine. But we also have some others. If you've taken psychology at all, you've probably heard of some of these. One of them is dopamine. Another one is called gamma-aminobutyric acid, or GABA. And serotonin, which I know you've heard about in psychology class. Dopamine controls our muscle movement. Serotonin controls our sleep and wake patterns that we have. We also have two gases, nitric oxide and carbon monoxide. And that's what you want to mark with these two, is that they are two neurotransmitters that exist in the blood as a gas. Now, acetylcholine and norepinephrine are what we call excitatory neurotransmitters. In other words, they encourage the postsynaptic neuron to fire. Whereas dopamine, GABA, and serotonin are inhibitory. In other words, they prevent the postsynaptic neuron from firing. And as you'll see in the video clip on the synapse that is actually on my Moodle page, it's the combination of those that actually determine whether or not a neuron is going to fire. So now we've gone through how an impulse is created, how it moves down the axon, and how it moves from cell to cell. So now what we're going to do in the next clip is to actually take a look at the difference between a typical nerve pathway, which we've kind of discussed, and a different type of pathway called a reflex.